So this is the answer booklet for Disease Defence and Treatment 4.6 Additional Biology. Again, you have the knowledge organiser here, which you should have used to help you answer the questions. Some different types of cells are listed below. From the list above, name 2 types of cells which are microorganisms, and the answer is bacteria and yeast. Use numbers from the list above to complete the table below. You may use the numbers once, more than once. First row has been done for you. Has chloroplast, 3 and 5. Has a cell wall, which is 1 a bacterial cell, 2 a yeast, 4 plants. Well, that should be 3 for a plant. Sorry guys. 3 for a plant, and then 5 algal. Animal cells don't have a cell wall. Has a nucleus, so 2, 3, 4 and 5. Bacterial cells don't have a nucleus, they have a plasmid. Has cytoplasm but no nucleus, which is our bacterial cell. The diagram shows a virus. Complete label A is our protein coat. How does a virus reproduce? Underline one answer from the 3 choices below. It multiplies inside a host cell. It can't do it unless it's in a host. The diagram below shows a section through the skin after it's just been cut by a sharp object. Explain how some other parts of the blood shown in the diagram can help protect the body from infection at the site of a cut. So, 2 things we're going to look here. We have platelets and we have white blood cells, are 2 we need to talk about. So, we can say the white blood cells defend the body from infection, but they generally do. There are 2 types. We have phagocytes and lymphocytes, and then they do 2 different things. So the phagocytes engulf and destroy pathogens, whereas the lymphocytes produce antibodies to attach to the pathogen. Platelets then, our second type, is form clots to make scabs. So they form the clots and it prevents blood loss and prevents microorganisms from entering. The diagram below shows stated in response to 1 type of white blood cell to a flu virus vaccination. Name the type of white blood cell that produces antibodies. The answer is lymphocytes. Describe the process that has taken place between stages 2 and 3. So between stage 2, I've got 1 lymphocyte, then I have many. So the lymphocytes have duplicated many, many times. State the importance of stage 3 in protecting the body from a flu virus. So the more antibodies produced, we get a quicker response. New forms of flu virus appear almost every year in the UK. Suggests why a government report recommends that flu vaccines should be given every year. It's because we have different antigens as it mutates, so different antibodies are needed to fight off that pathogen. Name the English doctor who first used vaccinations. It is Jenner. Remember, don't put the first name in case you get the first name wrong, and then you will lose the mark. After an injection, blood clots at the site of the wound. Suggests why it's important for blood clots. Well, it prevents blood loss and it prevents pathogens from entering. Immunity against polio is created by a series of vaccinations. First is given at age 3 months and is followed up by a series of booster vaccinations. The following graph gives information about these vaccinations. Use a graph and your knowledge to state the age range during which a person is most likely to suffer. Well, there's a dotted line that says the minimum level needed for immunity. So if you're below that level, you're more than likely going to suffer polio. So it's between age 0 and 1 years of age. Describe how the immune system reacts to booster vaccinations. The body contains memory cells that recognise foreign antigens, causes the immune response from lymphocytes producing antibodies, and it's much quicker. Smallpox is caused by a virus. Due to worldwide vaccination, it's now considered to be extinct. Name the doctor who used vaccinations. Jenner. As you can see similarities, they constantly ask this. Suggest why the flu virus has not become extinct even though vaccines are used to prevent it. It's because it mutates rapidly. Alexander Fleming found a fungus called Penicillin in a petri dish containing a culture of bacteria growing on agar jelly. The diagram shows what he observed. The first thing he observed was a chemical released from penicillium as harming the bacteria, and the chemical was diffusion through the jelly. What is the evidence in the diagram for each conclusion? For conclusion 1, that it's releasing penicillium harming the bacteria chemical, is that no bacteria grew around the penicillium, which you can see. The 2, was that it was diffusing through the jelly because bacteria started to grow further away from the penicillium. The chemical found in penicillin was extracted and is known as penicillin. What name is given to these types of drugs? They are antibiotics. Why has penicillin become less effective at killing bacteria in recent years? Because it's overuse of antibiotics and it's become resistant. MRSA has become a serious problem in hospitals. Describe one effect control measure used in hospitals against it. We can wash our hands, clean the walls. What term is used to describe bacteria which cannot be killed by antibiotics? They're resistant. How many doctors have made the problem MRSA worse through the overprescription of antibiotics? An influenza is caused by a virus. Why is it not possible to treat this illness with antibiotics? Because they only kill bacteria. Last question. The photograph below shows the badger. It is thought that badgers spread the disease TB to cattle. Badger culls aimed at reducing TB in cattle were planned for August. In certain areas, badgers would be shot. The cull would have tried to kill all badgers in each area. However, some scientists thought that a badger cull might actually spread bovine TB cattle onto other farms. Describe the possible link between badgers and bovine TB, that badgers spread the TB to cattle. The exact areas where the culls were kept secret suggest why is that people made protests to the culling of all badgers. A successful cull would kill all badgers in one farm. However, even if successful, a second cull would be needed after a few years. Why would it be needed? Because badgers from other farms that haven't been culled may migrate into those farms. Suggest one reason why scientists think badger cull might spread TB or to cattle on other farms. Because if not all of the badgers were killed in the cull, some would obviously try to escape and go to other farms. Suggest two other ways, apart from killing badgers, which would prevent the spread of TB. We could vaccinate the cattle, or we could put the cattle in housing to protect them away from the badgers.